Hi, everyone. Thank you for joining us today. We're going to meet your presenters, and then we're going to do an introduction to the biological membranes experiment. And then we'll do a demonstration with Graphical Analysis Pro. We'll do Q&A throughout, and then later we'll also show you where to find that recording after today. So I'd like to introduce your main presenter, Sara. She has a PhD in zoology from Oregon State University and a postdoc in neuroscience from the University of Texas. And she's been a professor of biology for 15 years. So she has quite a bit of teaching experience behind her before joining Vernier. And also presenting today is Colleen. She'll be answering questions in the background and helping Sara along the way. She has a BS in biology from Truman State University and a master's in science education from Montana State. She taught 6 years in Houston, Texas, and she was a research scientist turned educator. So again, feel free to ask all of your questions. We'd love to answer those. We'll be going over the features of graphical analysis. I'll put in the chat where you can find the link to download that here in a moment. I will pass it on to Sara.
All right. Well, hi there. Thank you for joining us. The lab that we're going to talk about today is from our Biology with Vernier book, and it's lab number 9. And the topic is cell membranes. And one of the challenges of teaching cell membranes is that most of the time, the options that you have might be somewhat limited. One of the most common preps that I've seen for many years now is using dialysis tubing. I think people have a relationship with dialysis tubing that's either love or hate. And so this prep is actually quite different. And I have found it to be very engaging with students. We're going to look at membrane properties using a model system with a vegetable. So just to kind of introduce the idea of cell membranes, just to remind everybody, they are composed of a phospholipid bilayer, and they have hydrophilic heads that face outward and inward into the cell. And then they have hydrophobic tails that are sandwiched in between. And that makes them semi-permeable. So only certain things can actually move through the membrane. So water and some smaller lipids can pass through pretty easily, but things like ions and larger molecules can't get through easily. So we are going to use, actually, Angie, if you would go to the next slide, we're going to use beets as a model system today. And the reason that we're going to do that is that beet cells contain pigments called betalains. And there are 2 main types. There are beta-cyanins, which tend to be in the red range in color, red to purple. And then also beta-xanthins. And beta-xanthins tend to be yellow in color through most of the pH range, or at least through the middle of the pH range. So we're going to actually focus today on beta-cyanins and using them as an indicator of how much the cell membrane is disrupted. So you can take a cube of beet. We're going to go through that today. You can incubate it in a different type of solution. We can test a bunch of different parameters. And then as the cell membranes are disrupted, you see more pigment leaking out. So you basically get a darker, more concentrated color of the pigment in the solution that it's incubating in. So if we measure that on a colorimeter or a spectrometer, you can actually get an indication, at least, of the relative amount of cell damage that's happening or membrane disruption that's happening with your absorbance readings. All right, so the way that we're going to run it today is we're going to do the main prep out of Biology with Vernier, lab number 9, and we're going to use the GoDirect colorimeter. Now, we do have other versions of this lab. We do have a version of this lab that is in the Investigating Biology with Vernier book, Investigating Biology Through Inquiry. That's a more, or I should say less structured way of doing it with students that gives the students the basic protocol. They can also, though, do inquiry-led experiments using this protocol from Biology with Vernier. It just gives them a little bit more guidance as they go through the instructions. and we're going to use the Graphical Analysis Pro app today. All right, so Vernier Software and Technologies main focus is software and technology that is designed for teachers. And most of us in my department are actually former teachers. As Angie mentioned, I'm a former college professor. Colleen is a former high school teacher. Almost everyone within our area and all of our biologists are all former classroom teachers. So we've used this equipment for many years, and we try to design experiments that we think that students and faculty will really enjoy. So we're going to use Vernier Graphical Analysis Pro for this demo, and we're going to actually do the measurements live. I mentioned we're going to use the GoDirect colorimeter. I should mention that you can do this lab with a spectrometer. You would just use a different piece of software, which is called Spectral Analysis. We'll talk about that towards the end. We'll go and revisit that using a spectrometer. But for today, we're going to use Graphical Analysis Pro with the colorimeter. And we're going to use the colorimeter can measure light absorption or transmittance of a liquid sample at 4 different wavelengths. So it's got 4 different essentially channels that you can use. We've got 430 nanometers, we've got 470. That's the one that we're going to actually use today. We have 565 and we also have 635. So we have a nice range of wavelengths that you can use on the colorimeter. All right, so without further ado, I'm going to actually show you how this lab works. So I mentioned that we're going to use beat, and I've got my colorimeter here in view. Let me switch out. I'm going to just show you very briefly what it kind of looks like when you're cutting these beat samples. So the idea is you want a piece, oops, let me switch our scale over here. We want to get a piece of beat that is approximately 0.7 centimeters, so 7 millimeters. And after a while, you get, and you want that in all dimensions, after a while, you get kind of good at gauging the size of your piece. I also, I personally like to have a balance nearby so that I can measure the weight of the pieces to make sure that I'm getting roughly the same size of a cube of beet. But it's something that if you're going to do it in lab, you might want to practice. You could certainly have your students cut it as well, depending on how much you trust your students with sharp objects like knives. I do recommend using a very sharp knife before you start. And I'm also going to, I just realized I should be putting on my PPE. This lab doesn't use particularly harmful chemicals, but you certainly don't want students splashing anything in their eye. The other thing to consider is that beet juice does stain. So we recommend with this lab that students use aprons and that they wear eye protection. So I'm going to put my eye protection on before I start handling any liquid samples. And I'm going to switch out here and show you what it looks like to set them up. Now, I kind of prepared this like a cooking show where I've got some samples that are already ready to go. So I started some of these samples already in a micro well plate. And those are the ones that you see along here. So normally you would just drop in a cube of beet into each well, and you would add your solution. Some micro well plates are a little easier to label than others. I find that I just have to remember where this is one of the ones that the wells don't have a surface next to them that you can write on easily. But you incubate your samples in your solutions. I'm starting, by the way, with a salt solution that ranges from 0% sodium chloride to 15% sodium chloride. And you can see that these have been incubating for about 10 minutes, and you can see the color has already changed in our samples. So what I'm going to move to is actually, we can start running our samples in graphical analysis in just a moment, but I'm first going to prepare our cuvettes. So you want to pipette the liquid in which your sample has been incubating into a clean cuvette. And then I prefer to put a cap on my cuvettes. And this is the 0% sample. Now you can have your students, in order to save cuvettes, you can certainly have your students drawing the fluid in and out of the same cuvette. That's perfectly fine. For our purposes, I'm just going to prepare separate cuvettes today. As we do this, bear with me, this is my 3% sample. And if you have any questions at this point, feel free to drop them into the chat. I know that Angie and Colleen are monitoring the chat. So the first thing that we're looking at today is the osmotic effect of different solutions on the plasma membrane. No questions yet?
Nothing yet.
All right. So this prep, what's nice about this prep is that students can really explore, if you wanted, I'm using sodium chloride, but students could certainly explore different types of salts if you have them available in your lab. I just used plain table salt for this prep today, which is the one that the main prep that's in the book. But you could certainly have them try out different types of salts. You have a lot of freedom with inquiry in this lab if students would like to design their own experiments. All right. And I'm going to show you a number of different explorations that you can do with this lab today. All right, so I'm going to switch views and I'm going to actually get my colorimeter ready to go here. That means I need to launch graphical analysis. So I'm going to start the graphical analysis software. Right, I'm going to change my view here so that you are seeing graphical analysis. So when you first launch it, you get a view that looks like this with kind of a welcome screen. We're going to pick sensor data collection, and we are going to connect wirelessly. So I'm going to connect via Bluetooth to this sensor. You could also just plug it in on USB. And Graphical Analysis Pro is a software that's available for 5 different platforms. So Mac, PC, Chromebook, Android, and iOS devices, which means that just about anything your students bring into the classroom, you can run the software on. And it looks pretty much the same on all devices. There are 5 different versions, so you'd want to make sure that your students get the correct version from our website. Our website has links for all 5 versions. All right, so we've connected our GoDirect colorimeter. The first thing we do want to do is calibrate it. So on the device itself, it has built-in calibration. So I am going to select the wavelength that we're using today. And remember, we're using 470. So I'm going to do that using these directional arrows. And then I'm going to calibrate using a blank sample. place it in the chamber, and then simply hit Cal. And if you're looking at my view as well, I know that the screen is graphical analysis. If you happen to have the members view also available in a sidebar, you're looking at the colorimeter.
Sarah, what solution are you using for your blank?
I am using plain water, just distilled water as my blank. All right. The next thing I want to do is I want to change modes because we're going to be looking at a concentration-based experiment. And I want to make sure that we're not trying to continuously collect data between samples. So we're going to use what's something called an event-based mode. So I went down to the bottom lower left corner and I selected mode. And instead of time-based, we're going to use event-based. And we're going to use something called events with entry. For our event name, we're going to change to concentration. And then for units, we're going to use percent. And then we're going to click done. And then we're ready to actually collect some data. So we're going to start with our first sample, which was just incubated in plain water. So 0% sodium chloride. And we're going to start collecting, but note that we don't have any points appearing yet. We've hit collect, but now our button choices are stop and keep. And what we want to do is when we are ready for a sample to be collected, that's when we're actually going to hit keep. So we want to watch our absorbance stabilize. You can see your absorbance in this lower corner. We can also put a meter on the screen to make it a little bit more visible. And when we're ready, we will hit keep for our sample that's in the colorimeter. And since this is 0% sodium chloride, we'll keep that point. And we're getting simultaneously transmittance and absorbance in our readings. All right, so now we can switch out our sample. Each time you put a cuvette into the colorimeter, you want to make sure that the clear sides are aligned with the lamp, and there is an arrow that shows you the direction of the light that's shining through. You also want to make sure that you always close the door on top before taking a reading. All right, we'll get our next sample. This one is a 3% sodium chloride solution. We'll move on to our 6%. We'll keep that one. And notice that each time we hit keep, it's plotting the results for us on the graph. Here's our next sample. This one is 9%. And we've got next one. This one is 12%. All right, and then finally, our last sample is 15%. All right, so let's take a look at our graph and we can talk a little bit about these results. They look like they're a little bit all over the place at the moment, but I will tell you that this is not terribly unusual. Typically you see the lowest reading somewhere in the 6% range. And so the 6% sodium chloride, what could we conclude if we end up, usually you get a little bit more of a U-shaped graph. But what could we conclude if we see this low absorption value for this middle range of solutions? Any ideas? It's okay if nobody wants to respond. I was going to say, if you want to put something into the chat, you're welcome to. But keep in mind that we started with our very, very low concentration. We've got probably what are hypotonic solutions with our cell. And then with the high end of values, we have what are probably hypertonic solutions, which means that in the middle, we're probably closer. Now, did we have somebody respond, Colleen?
Yes, Mary says isotonic point.
Excellent. Perfect. So we have found what is probably pretty close to the isotonic point for our beet cells. So you can play around with this prep a little. Certainly when you get a whole class of data, sometimes you get, you might get a more ideal looking graph that has a really nice U shape in it. But this prep is a great way to demonstrate for students that cells can be disrupted if they are placed into solutions that are osmotically imbalanced with them. Not quite as dramatic as something like putting blood cells into distilled water and watching them explode, but it is fun to be able to see the color change in the solutions and measure it with something like the colorimeter. I'm going to change now to, we're going to stop data collection, and I'm going to actually move to a different set of samples, because also in this lab, you can test detergents, and the effect of detergents on cells can be quite fun to look at. So I'm going to launch a new experiment. I'm going to go back to sensor data collection. And Colleen, remind me when we finish this set of samples, we can also demonstrate how we can live stream this data over the web. All right. So again, we're going to go into our event-based time mode, or sorry, our event-based mode. Once again, we're going to do concentration. This time I'm using detergent. I ended up using just basic dish detergent today. And what I did was I added numbers of drops to some water to dilute it. So my concentration here is actually going to be in unit of drops. All right, so once again, we're going to start with our 0 solution. You can see it's similar to our other experiment with the salt and absorbance value. Let me put the meter bar back up on the screen. It's a little bit easier to visualize that. All right, so that's our solution that was just distilled water. All right, and we'll switch to our next sample. This was just a single drop of dish soap in our solution. You can see already the absorbance really has increased. So just a little bit of soap. And this was number of drops in 10 milliliters of water. Got a nice increase in color. Our next sample is 5 drops. And you could certainly have students test anything from different brands of detergents. If you have pure sodium lauryl sulfate, you can mix up a known concentration. but you can also have students test household products. So things like laundry detergent or different types of dish soap. And here's our final sample. This one is 10 drops of detergent. And for an experiment like this, looking at detergent, the relationship between detergent and disruption of cell membranes, typically you get much more linear looking data. So I can show you a cool analysis feature in the software where we can plot, we've got the graph plotted, but we can now apply a curve fit to those data. And this looks fairly linear. So you can look at relationships between parameters as you use this sensor. All right, so I've done an analysis here. What I'd like to do now is, let's say that we're using the software in a scenario with remote teaching. If I had students that were not in the classroom with me right now, but I wanted to be able to do a live experiment and have them be able to collect the data as well, I can data share with them over the web. And this is one of the really neat features of Graphical Analysis Pro, is creating a live session that we can share with students. So I'm going to hit this little, it looks like this is a radio button up here, and that is the data sharing button. So it's kind of an old time radio symbol. And I'm going to start a session. So I have a code here, and what I'm going to do is I'm going to show this code, and anybody that wants to follow along can. I know that Colleen is planning to follow along. So Angie, would you mind dropping that code into the chat for anybody that would like to input this? And then Colleen's going to show you from her view as the student where students would put that code. So as the instructor, this is what my view would look like. Colleen, are you ready? I can stop sharing.
Yep.
All right, excellent.
Stop sharing and I will start sharing. So this is kind of how a student would be able to get this. And what's great is, like Sarah said, you can use this for any remote situations. You can also just use this within the classroom too, if you're just wanting to do like a demonstration and share the students the data. So I opened Graphical Analysis, and I am going to click on data sharing. And so for data sharing, we're doing online data sharing through the internet. And I'm just going to type the code that was displayed. So WDU0RS, and then connect. And it can take a couple seconds, but what's going to happen now is the data that Sarah had collected is now on my screen. So I now have the ability to look at this data, and you'll notice the big difference is that Sarah had that linear fit on her data. I do not have that. It did not carry over. So basically, whenever you have a sharing instance, it requires the students to then do their own analysis. So each one would be responsible for figuring out what the line of best fit would be for that. So again, I just go to Graph Tools, apply Curve Fit, and now I have my curve fit present there too.
Thank you, Colleen.
Yeah.
So this is you can see this is a pretty nice tool. Colleen and I are actually across town from one another. Actually, I think Colleen is technically in a different town. I am in Beaverton, Oregon.
I'm in Hillsboro.
So we are sharing this, and it doesn't matter how many people you connect to. You can have unlimited numbers of students connecting with you. It doesn't matter how far away they are. They can connect as long as they have an internet connection. They can get those data, and then they would want to save them, for instance, on their own device. And that way they can analyze them at home. So this is a really nice feature of Graphical Analysis Pro that is particularly powerful in a time, especially now, where we don't know sometimes if we're going to be teaching remotely or in person. But Colleen also mentioned that you can also do a local data sharing. So if you want to stream this out in class, or have, for instance, one group collect data on one device, but then share it with their group members, they could do this as well. So that each student, if you're one-to-one at your school, each student could still get a copy of the data, even if they only had one colorimeter. They can share from one central device out to the others. All right, any questions?
Mary was asking about the code for the app.
Ah, okay. So the code
Stop
is
sharing.
the code is generated with each data sharing session, and that is something that's important to remember. Is that and this may not have been Mary's question. I didn't see Mary's question, but each time a professor starts a data sharing session, you're going to see a unique code pop up. So you would not want to use that same data sharing session code for multiple sections.
She's talking access to the app.
Aha, okay. Let's go back. Angie, do you mind going back into the slides and we can talk a little bit more about the app? All right, so the software allows you to do either in person, obviously, or you can data share. The code to use the Pro version, we actually have provided in the webinar, and Angie has just put it into the chat. So this is the code to actually upgrade your version to, so the installer is the same for the free graphical analysis version and the Graphical Analysis Pro version. So once you install the free version, then you would actually use the code that's there on the screen or also in the chat. You would use that to upgrade your version. So if anybody has Graphical Analysis launched right now, you would want to look up on the top bar towards the right, and you might have a little shield with a check mark in it. Go ahead and click on that. That should give you a prompt to enter the code to upgrade to Graphical Analysis Pro. This code is for you to use. It should last for 2 weeks.
It is good till September 9th.
Excellent. All right. Thank you. And if you haven't tried Graphical Analysis Pro yet, go ahead and use this link today. It is going to be good until September 9th. And then you still could sign up if you want a little bit more time to try it. You still could sign up for a free trial. And the free trial is available on the website. If you, let's see, we can, Angie, if you give me control back, I can share, I'm going to share my screen. Put it back on me for a second here. I'll show everybody where to find it. All right. Let me share my screen. I guess I can probably take my goggles off at this point. All right, you should be seeing my view of the website. So if we go to products and then to software, or we could go straight to downloads. We can navigate here to Graphical Analysis Pro. And this is where you would find it if you were looking to purchase it. But there's also this live link here that says educators sign up to request a free 30-day trial. And that's where you would put in all of your information. The more information you give us, the better. Because we do, I believe, limit this trial to people who are actually teaching. So it's not generally available to the general public. But put in all of your information, your role, your school, and then let us know what you teach. And you can also opt in or out of receiving emails. We do try not to spam people. So we try to limit our email communication with you to subjects that we think you would be interested in, but you can opt in or out of that and give us information about what you teach. And then you can have the free 30-day trial. What I would do is try using the code that we gave you today. Go ahead and use that for the next couple of weeks. Because you don't want to activate the code, the 30-day trial right away. If you want to get a little bit more time to play with it and try it out with maybe even with your students in the classroom.
And Mary had a really good question. She asked if this will still work with her LabQuest 2 and LabQuest sensors, and the answer is yes, it will. You do not have to have Go Direct sensors for this to work.
Yeah, you can do exactly the same lab. In fact, the books, all of our books, the Biology with Vernier book in particular has different versions of each lab in the electronic resources. And the electronic resources are a fantastic place to find folders. So when you download the book electronically, you'll find folders for each major software platform that we have. So there are instructions for Logger Pro. So if you're a Logger Pro user, you could also use this lab, and that's actually the printed version that's in the book. But you could also find the folder for graphical analysis. You can find a folder for LabQuest. So you've got all of the different options for finding the student instructions that match your device. And that way your students aren't trying to figure out how to use instructions for the wrong type of software. All right. So I was going to show you if we have any more questions, we can handle some of them now. If we don't have a lot of questions, I was actually going to show you a cool version of this lab that's actually from a different lab, but it's the exact same basic setup, and it's the effect of alcohol on membranes. And I can demo that as well if you're interested. So Colleen, did we have any other questions?
Mary was asking if the LabQuest can transmit to GA Pro, and the answer is yes.
Yeah, and there's even an online, a data sharing mode that you can use for that as well over Wi-Fi. All right. All right, so I'm going to show briefly the effect of alcohol on membranes, which is actually lab 8 out of the Biology with Vernier book, but the results are always kind of fun for this lab exercise. Feel free to keep asking questions as I do this. This will not take terribly long because the experiment, I've just got one set of samples that are with isopropyl, looking at isopropyl on membranes. All right, now I'm going to share my screen again so you can see graphical analysis. All right, again, we're going to do sensor data collection. Now, Colleen, I believe, is getting a message that said that I just started a new experiment. Is that correct, Colleen?
Yes.
Okay, so in the teacher mode up here, I'm still in a live data sharing session, and I can see that I have 2 clients connected. This is something also that's worth mentioning, is that you can't see the identity of who's connected, but you can see how many are connected. So if you're worried about whether your students are connecting, you can't see each individual student as their identity. So there is some privacy here, but you can at least see, for instance, if your whole class is connected. All right, so again, I'm going to go into that events with entry mode. I'm going to use concentration. I'm using isopropyl, and it's again in a % solution. All right. There we go. Okay, so there's my first sample, and this is my 0. All right, so we're going to put in our concentration, keep our point, switch out. I'll do this fairly quickly. This is 10% isopropyl. And then 20%. Give it a second to stabilize. So as I mentioned, this is actually just the lab that appears right before the lab on osmolarity and pH. The one that I didn't demo today was pH, but the results are nice. You can also find the pH range. You're going to see more U-shaped data for the pH test. It's going to be a little bit more similar to the salt. And you'll see more linear data when you look at things like alcohol or detergents. All right. So those were our samples for our isopropyl. And you can see if we wanted to just look at this relationship here, it gets fairly linear once you hit the 10%. So, all right. So your students can really explore, you can have them try different types of alcohols. They can try methanol, ethanol, isopropyl, anything that you have available to them in your lab. And then of course I mentioned with salts, you can try different different types of salt, as well as different concentrations of salts. You can try different pH ranges. There are instructions in the lab for testing pH range. I'm going to stop sharing and come back to a view. I've taken my goggles off. So Colleen, do we have any more questions?
Not that I haven't answered already. It was, we did have one who didn't get the message that new data was collected and they had to do a new experiment and then use the code again.
Ah, okay. Okay. All right. Let's see. Well, trying to think of anything else to tell you about or share with you. As you can see, it's a fairly easy prep. Sometimes I think this one looks a little bit intimidating because you've got to chop up the beets, and that part of the lab probably is the longest, with having, especially if you're going to have students do their own prep. If you pre-cut things before lab, that really speeds the prep time up. One thing I didn't mention was that before you add the pieces of beet into the wells of your plate, of your incubation plate, you do want to rinse them very, very gently 3 times with a little water, and then draw the solution off. And that's just to make sure that you don't have any excess beet juice that's leaked out of the cells. You want them kind of clean going in, so that what you're really seeing with color change is truly the disruption of the membranes as it sits in the solution. Another thing to note, a common pitfall, is the cuts should be very, very neat and clean. You don't want any ragged edges or pieces that add more surface area to your cubes of beet. You want to try to get them as cube-like as possible and as precise in the cuts. That said, as long as your consistency, even if they run a little bit larger or a little bit smaller, as long as they're the same size, a consistent size, the lab should be pretty successful.
Oh, Mary had a great idea for an extension on this, of asking students to make a solution to clean a stain out of clothes.
Oh, that's great. That's a great idea. I mean, it is a great point to talk about with students as to why we use detergents, because most students don't necessarily understand the difference between something like a simple soap that emulsifies things like fats, and a detergent that actually can get in and disrupt proteins as well as cell membranes. So that's a fantastic idea.
Also, I'm not sure if you mentioned this, but I found when I've done this lab, you want to make sure you're not using any part of the beet that has skin on it. So
Yes.
you want to make sure it's all basically interior beet.
Yeah, I usually make a slice through the beet, and then I start cutting slices that are roughly the same width. And then I trim off any edges that have skin or root on them. And then I just cut from the interior of the beet. And I would probably not cut more than maybe an hour before your class time. I would try to like, I wouldn't let them dry out if you can help it. And you don't want to store them soaking in solution. So that ideally they are cut fresh, not too long before lab began, and are ready for students to use. And of course, your students, again, if you're willing to have your students cut them, they can also do their preparation themselves. So I mentioned earlier that you can do these labs also with a spectrometer. And if you were to do that, you would want to, we can just briefly show the difference. The GoDirect SpectroVis Plus can be used for this lab. And if you do have a spectrometer, then you may already be aware that graphical analysis does not work with spectrometers. For that, we have a different software, and that other software is called Spectral Analysis. And I'm gonna launch that in a moment here. And I'll just show you, there are some basic similarities, but I will note that spectral analysis is not going to give you the options of doing things like the live data sharing over the web. So I'm going to share my screen just to show you spectral analysis. So when you launch, you get a very similar general kind of the launch screen. We do want to connect a spectrometer, and to do that, we want to turn on our spec, which seems to have, in the time that I sat here, seems to have run out of battery. It hasn't been charged recently, so we'll see if we can get it plugged in. But in spectral analysis, you have different options of your modes. So you've got the option to look at the full spectrum of wavelengths. You've also got the option to look at a value of absorbance versus concentration. This would be what you'd use for things like a lab on Beer's Law, but it's also the mode that you would use for this particular lab. And you're going to notice that you would notice if you're running this, that the interface looks a little bit like it did for graphical analysis, where you are prompted to put in a value for your concentration with each sample that you take. And then because I don't have that spec connected, it's not letting me show you the next screen, but we're starting to run a little bit low on time. Well, I hope that you will try this lab out. I have always found this one to be a bit more engaging with students than doing the dialysis tubing, and I will say it's a lot less messy. Even with the beets being red and staining things, you will probably find that there's a lot less cleanup for this lab than there is if you're doing a lab with dialysis tubing.
Yeah.
Ways to get a hold of us if you need us. Colleen and I are 2/3 of the biology department. So if you contact us, you are most likely going to get one or the other of us. Also our director, John Melville, he's also a former biology professor who taught at the college level. So all three of us have lots of teaching experience. And we can be reached if you want to put a question into email. We have an email that is support@vernier.com. If it's a biology specific question, then you can actually get kind of a straight line straight to us. And again, there's 3 of us. So we're not a huge company, so you're not going to get some random person. The biology team, you can specifically email us at biology@vernier.com, and that's a great way to get a hold of us. Also, if it's within our business hours, we are on the West Coast, so we start answering phones usually around 6:30 in the morning. We also start looking at chat somewhere between 6:30 and 7 a.m. Pacific time. And the phones are usually open until 5 Monday through Thursday, and then they close at 3 o'clock on Fridays. So we do have we close a little bit early on Fridays. So chat and phone runs with our business hours. Email tends to be answered also within those business hours, but sometimes you may be able to get messages after hours if you're lucky. So if there's anything else you need, let us know. But feel free to contact us. We love chatting with customers. So if you are curious about something or you wanted to clarify something, please let us know. Thank you for attending, everyone.